Good day, everyone. My name is Adhityo Wicaksono. Everyone call me Adit, and I'm from Indonesia. I've been working on Generasi Indonesia Foundation for several years behind, and I was assisting my colleague from Indonesia Institute of Science for doing one of the craziest plant research that I ever seen. Okay, about it, let's see. Let's go for the share screen now. Today we talk about Rafflesia. This is what the crazy plant that I talk about. This is a parasitic flower that is originally from Southeast Asia, but I will put the highlight here on a species that I found in Bogor Botanical Garden, Rafflesia padma. Just like the Rafflesia, this plant doesn't have any root, stem, and leaf. We can only see the flower that pop out from the host plant surface. Okay, before we start, I want to give so much credit for my colleague here, and basically also my research supervisor who have been that I've been helping for the last 5 years, Ibu Sofi Musidawati from Indonesian Institute of Science or LIPI, and she's also the part from Forum of Amorphophallus and Rafflesia Indonesia or Foramor, who has dedicated her 20 years studying this awesome flower. Okay, let's continue. If you want to know how many species are there are quite a lot. We have 41 species in total based on the data from Plant of the World Online of the Kew Garden. You can access it online. All of these species are scattered from the peninsular Malaysia, Sumatra, Java, Borneo, and Kalimantan, and up to the Philippines. And all the species scattered around this area are come with their own uniqueness. As for example, if I remember, what in Philippines have a very unique form of the polygonal tube, the one in the middle, if you can see, the one that resembles the part of the eye. It has a certain uniqueness with the species in the Philippines. Okay, as we can see here, perhaps we can see now that Rafflesia is indeed a flower, as you can see here. But if we look again, there are many unfamiliar organs that we can never see in the normal flower. For example, instead of having a distinguishable petal and sepal, it has perigone. Then the stigma, here you can see the perigone is the one that appears like a petal, but it's not actually. Then the stigma and anterior region is located hidden inside the central disc. Almost all of the Rafflesia is dioecious, which means it has a separate male and female flower. Although there are two species from Philippines, Rafflesia balletii and Rafflesia verrucosa, which are bisexual species. Okay, and to note, the surface of Rafflesia is leathery. It tends to mimic the shape of the skin of the mammal. And when I see closer to the flower, it doesn't have any phloem tissue on their vasculature. It only has a xylem and a vascular parenchyma to aid the nutrient distribution. On the right here, the seed is similarly strange. It has no cotyledon, only a strange embryonic tissue. We call it a Brownian nucleus, Brownian nuclei, if I remember. In case of bilobe seed that resembles a nut with a really strong integument layer. In terms of the flower growth, so far we discovered that it has either aerial, as it grows, an exposed root in the host plant, and the terrestrial one, if we can see here on the right, that grows on the ground level root. But most of the Rafflesia species are terrestrial, as the aerial believed to have more difficulties to obtain nutrients as it's far above the ground, hence the flower bud mortality is really high. So far as I can understand, only a Rafflesia species with small flower that support most of the aerial flower. Okay, let's continue. Now we talk about its life cycle. Until the moment of dispersation being made, we have no idea. We have not yet acquired the whole life cycle from its seed germination to the point of the fruit formation. None has seen how the seed germinates, But we know that the later stage include the parasitic tissue and our endophyte grows inside the houseplant fruit vine. That stage, the tissue is completely hidden for years until it decides to grow a flower bud. So yeah, it completely hidden inside the host plant. Here, in our 2019 paper, we found the cell cluster that we believe to be the endophyte. According to the paper from Nikolov et al. 2014, it is distinguishable as the parasitic cells have a larger nuclei. It means it has a big. It's assumed to be have a greater genome, and it means that it also divides really slowly compared to the cell outside the endophyte tissue, the host tissue. As we can see here, they reside in the host vascular cambium. The one marked with CA is a cambium here. We believe that it can spread as the cambium grows by the force of the dividing cell around it. But to make understand it clearly and to prove it even further, we have to do more investigation for this. Once the endophyte decide growth of flower bud, it grows from the host vascular cambium layer outward to the periderm, the cork layer of the host, until it bursts and form a flower bud cupule that initially swells on the host surface, like the picture on the right here. Sorry, I will move this one first. Now you can see. As it grows larger and larger, it also alters the host vasculature. As you can see, there's a high HAX here is the host anomaly cell xylem, which is become bent due to the growth of the Rafflesia parasitic tissue here. And the phloem, the HSP, become even more bent as it become pressed sideways. Compared to the one here, the one in the bottom, as you can see in the bottom most of the right picture, it just grow completely normal without any bend in the vasculature. Now, once the cupule is getting larger, the Brack will reveal. The Brack is the one with the dark scale here. And much later, the Brack will upsize and it will show the perigon layer inside it before it blooms and the anthesis period starts. Now that the flower blooms, it only stays up to 5 days before it wilts. The flower emits rotten odor to attract carrier insect to help with pollination. And this type of pollination by the carrier insect is called sapromyophily. Other than odor, the flower appears to also generate heat according to Patino et al. 2002. So to enhance the flower capability outside of the leathery surface, it deceives the insect to believe that it is actually a dead mammal. Once the fly enter, it will be covered by sticky pollens, and then when it escape, the fly will come to the female flower pollinating it. And so the pollination, as the pollination start, it will turn into fruit in several months. Here is the fruit. I will move this back again. Sorry for this. Okay. The fruit is formed in up to 3 months. And strangely, the fruit can also form even there's no pollination. And this is called agamospermy. This statement was made by Dr. Jamli Nais on his book. But the reason, what the outcome is, the seed will be sterile compared to the one with pollinated one, which means it has potent and fertile to form a new individual of the Rafflesia. The fruit here resembles a woody cylindrical mass, and when open, there are a lot of crevices with thousands of seeds inside. The seeds are potentially spread by help of small mammal, like the squirrel and the trishu. I read the research from Dr. Ban Ziegler from Chiang Mai, Thailand, and it appears that the droppings from the tissues which has eaten the Rafflesia seeds and the fruits. It appears that the seed survived to the process in the shew's digestive tract, leading it to spread in any host plant that it found in the way. So that's the possible way to spread the Rafflesia seed naturally. Now we talk about the host plant here. From before, we only referred the host plant, but what is actually the host plant of Rafflesia? It is actually a vine from a grape family called Tetrastigma. It is a liana species, hence there is a debate if the aerial Rafflesia could actually grow on the Tetrastigma stem or it is an aerial root. So far, from what our recent study, we found that it actually grow on the aerial root exposed by the growth of the neighboring tree. So yeah, in the end, we believe so much that Rafflesia is a root parasite instead of stem parasite. Now we go to the past. Based on the study by Bendixby et al. 2010, it is believed that Rafflesia was originally came from Myanmar region before they spread to Malaysia, Indonesia, and then Philippines. Sapria was the first to separate. Sapria is the oldest of. We believe that Rafflesia is the oldest of Rafflesia. Say it also able to grow on the family, the genus of Brack, Vitis, instead of only in Tetrastigma. And then the Rhizantes will separate from the Rafflesia. Both Rhizantes and Rafflesia are only capable to live on the Tetrastigma instead of on the Vitis as well. However, the puzzle is now of how the parasitism start is still unclear, as well as how Rafflesia tissue evolved to be so minimalistic. We have no idea, and we still continue to investigate that. This is the oldest part now. By the locals in Malaysia and Indonesia as well, the dried Rafflesia bud is believed to cure postnatal bleeding and also to induce sexual vitality. But to answer that even better, we have to see to the scientific analysis on the compound, right? Yeah, the scientific studies have been conducted, and the studies revealed here that we have phenolics on alkaloids in Rafflesia, and also some more complex compound like gallotanin and syringin, which synthesize in the same metabolic pathway as lignin. Some studies also revealed the extract ability to heal wounds, as well to provide antioxidant and microbial properties. However, to get this potential, we have to focus on the propagation first. Without understanding on propagation, people will start harvesting Rafflesia without consent, and that's really dangerous for the ecosystem. Amazingly, the most successful way to propagate Rafflesia is by grafting the host plant, as my supervisor did on Bogor Botanical Garden. Some local claim that they also succeed by doing host vine cutting and seed germination. But we haven't yet documented it, and it requires more investigation and, of course, documentation. When we use the state-of-the-art biotechnology, like the tissue culture, which we can see in this picture, unfortunately, we brought more failure than success. The closest one to success was attempt by Dr. Sukamto and Mr. Mogiono in 2010 paper, which they claim to generate callus from Rafflesia tissue induced by synthetic auxin called picloram, which actually serve as a herbicidal compound. However, no organogenesis was found even even in the further induced with zeatin or kinetin, if I remember. Zeatin, the cytokinin. no morphogenesis was occurred. We are still currently still doing the research to see the effect of hormone to the bud tissue to further understand this. Okay, in terms of conservation, from before we learned that Rafflesia has a potential as medicine, right? It is also a good model for a parasitic plant study, and also it is a good part of the tourist attraction. Interestingly, education here will be required to be more part to be encouraged to more people, especially there are many people still believe that Rafflesia is a carnivorous plant. I met a lot of people like here like that, and it is also has to be protected from habitat destruction, as we haven't yet understand the life cycle. We need to learn from the locals that and have more study on the propagation. In this, when it is highly encouraged, because despite so far we see that Rafflesia is have a really complicated way to propagate. If that actually happen to be so, Rafflesia will be really hard and become really rare. But the fact is, the natural way to propagate the seed occur out there, and we believe that the propagation of Rafflesia is actually not really that complicated. We need to understand more about that. Yeah, I think, and we believe so much that the locals probably know better about it. There are still many mysteries for us to unravel from this alien flower. But the most important thing is to learn how we can propagate it to compensate its function, such as medicine. We need to conserve it is as become rarer and rarer. If we also want to do more research in the future and in less invasive ways, like in the silico approach, is greatly encouraged. And also metadata analysis and biomechanical modeling. I have a friend that told me that there's a way of the something similar like a CT scan that now can be done on plants. I think that's also a good idea to do it for Rafflesia. So in conclusion, Rafflesia is a unique parasitic plant. It has minimalistic tissue to thrive under its host. Its life cycle is still enigmatic. Also, its life greatly dependent on its host. As we see here, it has potential as medicine, but propagation study has to be understood more. First, and before we explore to the medicinal properties, we also need to do less destructive methods of analysis. And some Rafflesia species are endangered. We need to conserve it at all costs, so the next generation can learn its uniqueness and awesomeness. Don't you think so? Thank you very much for joining this presentation. If you have any question for or another queries, please don't hesitate to contact my email here. Hope this presentation will provide something new for you, and see you around. Bye-bye.